Dinesh? Dinesh, are you in line?
Yes, sir, I am in line.
Sir, 5 more minutes, sir. We'll start the session. HOD ma'am will come, sir. Hello?
Yeah, ma'am, sir is online. You can speak.
Dinesh?
Sir?
Dinesh, are you in line?
Yes, sir, I am in line.
Sir, 5 more minutes, sir. We'll start the session. HOD ma'am will come, sir. Hello? Hello? Hello?
Yeah, ma'am, sir is online. You can speak.
Yes. Sir, 5 minutes, sir. Ma'am will be online.
Screen ஆஃப் பண்ணிட்டாரு சொல்றாங்க.
Ajay.
Sir?
Dinesh, are you online?
Yes, sir. I'm in line.
HOD ma'am, are you online ma'am? Hello? Vasanthi ma'am, are you online ma'am?
Yes, Dhanalakshmi, I'm here. Can you hear me?
Yes, yes ma'am.
Yeah.
Yes ma'am, now I can hear you. Can we start ma'am?
Shall we start? Sir is there?
Hello? Yes ma'am. Yes ma'am, he's on there. Yes ma'am, we'll start ma'am.
Okay. All right. All right. Have you kindly see?
Sir, ma'am?
Please ask the participants to mute the
Participants, please. Participants, please mute your audio ma'am.
Yes, Dhanalakshmi, I'm here. Can you hear me?
Yes ma'am.
Shall we start?
Ma'am, yes ma'am, we'll start. Yes ma'am.
All right.
Yes ma'am, participants are there ma'am. Sir is also there. You can start ma'am.
Okay. All right. So shall we start now?
Yes ma'am.
Okay, all right. So, yes, we will start. All right. Good afternoon. All right. Yes, we will start. Good afternoon to everyone, and warm welcome to the webinar series in Animal Sciences 2. Before I proceed, can I ask all the participants to mute your audio? Please mute your audio. So we kindly request all the participants to mute your audio, otherwise it will be very difficult for you to listen to the resource person's talk. Yes, we will start. Good afternoon to everyone, and warm welcome to the webinar series in Animal Sciences 2. Before I proceed, can I ask all the participants to mute your audio? All right. Dhanalakshmi, am I audible now? Hello? Dhanalakshmi, am I audible?
Yes, madam.
Yes. This is Dr. Sandhanam. Is this Dr. Sandhanam?
Yes, madam. Good afternoon. Good afternoon.
Yes, good afternoon. Good afternoon, sir. So I cordially welcome all the participants for today's webinar series in Animal Sciences. And it's a pleasure that we have two very good resource persons, one from Bharatidasan University, Dr. Sandhanam, who is from the Department of Marine Sciences. And he's going to talk about recirculating aquaponics systems, which is something that takes off today in the field of aquaculture. And we are also very happy to have Dr. Dmitry Lajus from the St. Petersburg State University of Russia. It's indeed a pleasure having both a national speaker and an international speaker of repute. And we hope the participants have a very resourceful day today, and they are able to take back key factors that would help them in their own research. So now I will just hand over the floor to Dr. Dhanalakshmi, who will be actually gearing this webinar series for you today. So over to Dr. Dhanalakshmi.
Thank you, ma'am.
Yeah.
Good afternoon to one and all. It's a moment of great privilege and honor for me to extend a hearty welcome, big and warm enough to encompass you all, on behalf of PG and Research Department of Zoology for this one-day webinar series in Animal Science 2 on topic Challenges for Water Resource Sustenance. First of all, I take this opportunity to thank Lord Almighty for gracing this occasion. Small cheers and great welcome makes merry feast. I extend my welcome to our Honorable Secretary, Reverend Sister Dr. Ruby Alangarameri, and to our beloved Principal, Reverend Sister Dr. Helen, enthusiastic Vice Principal, Reverend Sister Dr. Mary Fabiola, our Head of the Department and Accuracy Coordinator, Dr. Pauline Vasanthi Joseph, my dear faculty members from other universities and colleges, and my dear colleagues, fellow friends and students. We are happy that at this point of time, we are optimistically engaged all of you, and we hope such webinars can help you actively resume back to research with fresh ideas. Every thought you produce, anything you say, any action you do, it bears your signature. According to this words, here we have Speaker of the Day, Dr. P. Santanam, Associate Professor, Department of Marine Science, Bharathidasan University, who is among us today to deliver a lecture on the topic Integrated Recirculating Aquaponic Systems, a new avenue towards the sustainable agriculture production and water conservation. Sir has graduated and received his degree, MSc, MPhil and PhD in Marine Biology from Center of Advanced Study in Marine Biology, Annamala University, Parangipete, Tamil Nadu. He has 14 years of teaching experience and around 20 years of research experience. Marine planktonology and aquaculture is his area of specialization. His core areas of research expertise include taxonomy, biodiversity, biology, biochemistry, ecology and ecotoxicology of marine plankton, nanobiotechnology and bioremediation efficacy of marine plankton, mass production and application of marine plankton in aquaculture, sustainable aquaculture practices including biofloc, copyfloc, aquaponics, RAS, and IMTA. Sir has guided four postdoctoral theses, 15 PhDs, 14 MPhil, 7 PG students, and 2 UG students. He is currently guiding 3 postdoctoral scientists, 6 PhD students, 1 MPhil, 6 PG, and 1 UG students. Sir has published 5 books, 4 manuals, 1 monograph, and 80 chapters in books and proceedings. Sir has published 246 research publications at the national and international level, with cumulative impact factors greater than 80, with H index 18, ITIN index 47, and total citation of about 1,297. Sir has developed 40 microalgae species, 2 rotifer species, 4 Artemia species, 3 cladocerans, 12 copypod species, and submitted 50 gene sequences in GenBank on microalgae, copypods, actinomycetes, and bacteria. Sir has educated 40 PhD theses, 20 MPhil, 40 MSc theses. Sir has organized various national conferences, seminars, workshops, and training programs, and has visited countries like Dubai, State of Eritrea, and Yemen. He has also participated in 145 conferences, seminars, workshops, and training programs, and delivered 99 invited lectures and participated in 27 training programs. He has also worked in various highly funded projects in association with the BBC, UGC, SERP, and has completed 7 projects and currently working with 2 highly funded projects associated with BBT, MOEF, and PC, with a total grant received about 477.87 lakhs. He has received many awards. To name few, Young Scientist Award by DST, Best Research Award by KTSTM Trust, and Professor Dr. M. Aruchalli Award. Sir is a member of 29 Academic and Scientific Committee and Advisor of Fisheries and Aquaculture, MSSRF Chennai, and Reliance Foundation Mumbai. He is an editorial board member in 5 national and international journals, reviewer in over than 25 national and international journals. He is the chairman for the Board of Examination of Nirmala University. Sir has done so many research developments and achievements. A name few is developed an RAS for large-scale culture of marine copyposts for the first time in India. He has developed immobilized microalgae photobioreactors for the first time in India. Established microalgae and copypost culture facility at the vicinity of the Bharathidasan University campus. In addition, he has published book titled Advances in Marine and Brackish Water Aquaculture. It's a Springer edited journal, book rated 45th place among the 57 best marine biology books. We welcome you, sir. We are pleased to have such a resourceful person with us today, and I welcome you, sir, on my behalf and my department and my colleagues. And now I request sir to take over the session, and I wish all the participants a resourceful day. Thank you. Over to you, sir.
Yes, ma'am. So I thank Dr. B. Dhanlakshmi, ma'am, who is an organizing secretary of this webinar, for having invited me to share my experience in the field of aquaponics. I hope you hearing me.
Yes, sir, we hear you, sir.
Okay. So, very good afternoon, one and all. Good afternoon to my professor and my well-wisher, Dr. Nadu, the professor from St. Petersburg University, Russia, and all teaching faculty, research scholars, and my dear student friends. Very good afternoon to one and all. It gives me an immense pleasure to meet you all through this webinar program. For this, I should thank the authorities of esteemed Nirmala College for Women, Coimbatore, India, for organizing an important webinar on an important topic, which is the need of the hour. So at the outset, I would like to thank the authorities of the Nirmala College for Women, the head of the department, and the organizing secretary, Dr. Dhanlakshmi ma'am, for having invited me to give talk in this webinar. Good afternoon, my good friend and my well-wisher, Dr. Edward Benjamin, sir, is a professor in the education department of our own university. So let me start today's lecture, entitled Integrated Recirculating Aquaponics System, a new avenue towards the sustainable aqua agriculture production and water conservation. I hope all the participants are getting the PowerPoint slide in our screen. Okay. So whenever I used to give the lecture like this, I keep this picture as a first slide. The reason, this is the one of the rare historical photo which was taken by a famous South African journalist, Mr. Kevin Carter, when he was visited Sudan in 1993. He took this picture. For this, he won a Best Photograph award. Okay. But after some years, the Kevin Carter, he committed suicide because of taking this picture. I will tell the secret behind, the reason behind this picture. So there is a girl child, she is attempting to move the UN camp where the moon meal is serving. And unfortunately, that girl child she cannot able to move further due to her malnutrition and health disorder. And you can see in the picture, there is a vulture is waiting for the girl to die and to eat her. So this is a real situation in all undeveloped countries and some developing countries. So the message, what I would like to tell by this picture, there are a lot of people, millions of people struggling for their food, for their nutrition, and due to their malnutrition, poverty and hunger, there are millions of people are dying every year. Okay, this is one of the critical situation in all the country, throughout the country, and most of the undeveloped countries and some developing countries. So here I have given some statistical data on global food crisis. Okay, so according to World Health Organization and Food and Agricultural Organization, they are stating that there are 113 million people across 53 countries experienced acute hunger. They are requiring urgent food, nutrition and livelihoods assistance. And the countries like Yemen, the Democratic Republic of the Congo, Afghanistan, Ethiopia, the Syrian Arab Republic, the Sudan, South Sudan and Northern Nigeria, these countries are facing a severe and acute food crisis. So these 8 countries accounted for 2/3 of the total number of people facing acute food insecurity, amounting to nearly 72 million people. Likewise, the food crisis is there. Similarly, there is a water crisis in the world. Very recent past, we are struggling. There are a lot of people are struggling to get the safe water for drinking and other purpose. So according to World Health Organization, there are 884 people across the world do not have access to safe water supply. There are over 840,000 people die every year due to water-related diseases. For example, cholera, jaundice, and typhoid. This might be due to the lack of clean drinking water. And almost 2 in 3 people who are deprived of safe drinking water. Across many developing countries, water is not available on land, but a daily struggle. The women are forced to work hours to secure water for their families, and the water they get access to is usually polluted. This is in turn leads to diseases and illness. The water contamination cause disorder on the fragile immune system of especially children. Due to inadequate clean drinking water, sanitation and hand hygiene, the diarrhea kills an estimated amount of 842,000 people across the globe every year. So now we are facing the pandemic COVID-19, but we have a historical data. Every year, there are 842,000 people are dying due to diarrhea because of lack of clean water for drinking and other domestic purpose. So over half of all primary schools in developing nations, especially in developing nation, over half of all primary schools, they do not have adequate water facility. Almost 2/3 they lack adequate sanitation. In the coming 8 years, 1.8 billion people will be living in regions with absolute water scarcity. And 2/3 of the global population could also face water stress conditions in the form of regular shortages and heavy contamination in water. So this is a real picture, the global scenario of food crisis and water crisis. Okay. So you may ask, what is the relevance of this data to the topic, today's topic? So we're going to discuss about how far this aquaponics is helping for water crisis or food crisis. Okay. And let me coming to the Indian scenario. So far we have discussed about the world scenario. So now I am coming to Indian scenario. Do you know there are 15% of the Indian population every night sleeps hungry. Till today, there are millions of people in India, they are surviving with one-time meal. So 194 million Indians are undernourished. There are out of 117 countries, India ranked at 104 at the Global Hunger Index. This issue, which is hardly finds its place in media reports, prime time, or in sensational or breaking news. This is a real situation in India. We are talking about this satellite and developing the other sectors. Okay, we are launching n number of satellites. We are spending millions of billions of money to develop and launch the satellite and develop some information technology and computer and other etc. But till today, there are 194 million Indians are undernourished and they are surviving with one-time meal. Likewise, even India, we are facing the water scarcity. India is at one of the country, actually according to World Health Organization and Food and Agriculture Organization, India is one of the country facing a major and most serious water crisis. Okay, there are 99 million people, they lack access to safe water in India. 541 million people, they lack access to improved sanitation. The India faces overwhelming water security challenges. More than 600 million people are facing an acute water shortage. About 3/4 households do not have a drinking water facility. See, the India ranks 128th among the 122 countries in the water quality index. So it is expected to be in 2030, the India's water demand is expected to be double to that of supply. That implies not only water scarcity for numerous people, but also a loss of around 6% to GDP. Our Indian GDP is going to drop at 6% GDP because of water scarcity. So this is a real situation in globally and India is concerned. Okay, now I am coming to the very important sector which provide food, employment, livelihood, and the income generation for the majority of people living in India. You can see India ranks second in the world in farm outputs. The agriculture production, milk production, animal husbandry and fisheries. So India is standing second place. So as per 2018, the agricultural employed more than 50% of the Indian workforce. And the agriculture sector means which include animal husbandry and fisheries, which contributed 17 to 18% to the country's GDP. According to latest report, the agriculture is primary source of livelihood for 58% of the population in India. So this statistical data clearly emphasizes that the agriculture sector is a dominant sector in India, which is providing employment and livelihood. In 2016, agriculture and allied sectors like animal husbandry, forestry, and fisheries, they accounted for 15.4% of the GDP of our country, our India. Even though our agriculture production is high, we are standing second in the global scenario in crop production, fisheries production, and animal husbandry, and etc. But till today, even though you can see here, I have listed out some national shame. Our former Prime Minister, Dr. Manmohan Singh, he stated that even though we are strong in different sectors like information technology, space, and so on, but still there are 70% of the children between the age of 6 months to 5 years are anemic. 20% of the children under 5 years age they are under acute undernutrition. And 48% of the children under chronic undernutrition. 28% of the men, 33% of the women, they are under malnutrition. And similarly, 24% of the men, 56% of the women, especially pregnant women, they are anemic, they are in anemia. So this is actually the real situation now we are facing in India. So millions of people, they are living in malnutrition, poverty and hunger state. So this definitely this will be considered being a national shame, as India is a global player. Okay. So if you ask me how to solve this malnutrition related issue, how to solve this malnutrition or hunger or poverty issues. So as a scientist, as a biologist, I can tell the only solution to solve the malnutrition related issue is nothing but agriculture and fisheries sector. Okay, because why I am telling the agriculture and the fisheries sector, the agricultural crop, grains, cereals and other grains as a good source of nutrition. Similarly, fish and fisheries product is a good source of protein and omega-3 fatty acids. So if you supply more agricultural crops, grains and so on, and fish products, so we can solve the malnutrition among the peoples. Unfortunately, so we are talking, the India is a second in crop production, but in recent 40 years, due to scarcity of water, due to rainfall, the failure in rainfall and the pollution, and due to climate change, because of these various reasons, now the agriculture and fishery sectors, they are facing threats. We are not able to grow the agricultural crops or fish or other food commodity because of lack of water. Because for the production of agricultural crops and fish, we need a large volume of water and more space for the production. So because of climate change or global warming and the failure in annual rainfall and pollution, because of these reasons, our agricultural sector is facing threats. So once the agriculture sector, the wild sector is facing threats, where we can get the nutritional food? Because already we are facing problem cultivating the crops. So the farming, when the wild resources depleting drastically. So according to various reports, the wild production and even the fish production in the wild is keep on declining drastically due to various reasons what I explained here. Okay. So the ultimate source to get the protein and nutrition is farming. The farming means aqua farming, agri farming. Okay, that is the only potential source which can supply the nutritional protein source to the poor people. Okay, but unfortunately, due to various reasons, for example, there are a lot of diseases. Even in agriculture, there are a lot of diseases are occurring, and even in aquaculture and fisheries, there are a number of diseases are reported every day. Okay, so the farmers are facing a problem to grow their agricultural crops and their fisheries crops due to diseases, and one side is disease, another side is water crisis. Okay, so to solve the disease-related issue, the farmers, agriculture farmers, they are using fertilizers, pesticides, insecticides and chemicals. And similarly, the fishery scientists, farmers, fish farmers, they are using tons and tons of chemicals and antibiotics. Just they are simply dumping the chemicals, fertilizers and pesticides to grow their crop, agricultural crop and aquaculture crop. And this application of fertilizers and antibiotics, that will give some appreciable number of percentage of yield, a good result in terms of yield and production. However, unfortunately, the chemicals, fertilizers pesticides, what the farmers are using in their agricultural land and aquaculture farm, it will become nowadays a major issue among the consumers. So whatever the chemicals or pesticides they are using to grow their plants or fish, that will be accumulated in the crop, that will be transferred to the end user, consumers. Consumer means human being, which will create some health risk, health disorders with the consumer. So because of this reason, now in aquaculture and even in agriculture, some pesticides and some chemicals are banned by Government of India, so that the farmers, they move towards the organic farming. Organic farming means using the organic manure or organic fertilizer. So by using the organic fertilizer or organic manure as a fertilizer for the agriculture production and also the aquaculture production, so the farmers, they are moving towards the organic farming. Okay, the organic farming is nothing but using the organic waste or organic manure, a good source of nutrition to the agricultural crop production and aquaculture production. Again, unfortunately, they left out organic waste, the organic manure, and unutilized feed, and the fecal matter, the excreta released by aquaculture organisms. So they left out organic waste, again will create unwanted environmental issue. The environmental issue is nothing but eutrophication. So this organic manure, they left out organic feed and organic manure, it will create another unwanted water quality problem. So the water body, all water bodies, wherever the agriculture farming is doing, wherever the fisheries or aquaculture farming is doing, that all wastewater which is discharged from the aquaculture and agriculture land, when they reach the water bodies like a sea, ocean or estuary or river, it will pollute the water body. Again, this will be another headache among the environmentalists. Okay. So, with this, okay, so the eutrophication and algal bloom is another unwanted issue nowadays in the environmental field. Okay. So now here I have given some statistical data about Indian aquaculture. As we discussed earlier, India is the world's largest producer of milk, pulses and spices, and even the cattle, and even in fish. So India is the second largest producer of rice, wheat, cotton, sugarcane, farmed fish, sheep, goat, meat, fruit, vegetable, and etc. Okay, the country has some 195 million hectare under cultivation, of which some 63% are rain-fed. You just please note down this, you underline this sentence. 63% are rain-fed, while 37% are irrigated. So as we know, we are all well known that due to climate change or global warming, now there is a drastic drought. There is a failure in annual rainfall or monsoon rainfall. Okay. See, nearly 3/4 of India's families depend on rural income. The majority of India's poor, there are, some 770 million people, or about 70% are found in rural areas. Why I am telling this rural area means, rural area is an area where the agriculture and aquaculture or fisheries farming is going on. Okay, so India's food security depends on producing the crops as well as fruits, vegetables, milk and fish to meet the demands of growing population with the rising incomes. Okay, so this so far we discussed the agriculture and the fisheries or aquaculture is a major source of nutrition and protein for the human being throughout the world, and India is also in particular. Okay. As you know well, water is a major medium to grow any crop production. Okay. For agriculture, we need an enormous volume of water. For aquaculture and fisheries, we need a large volume of water. But due to failure in monsoon, the rainfall, where you can get the water for agriculture production and aquaculture production. Okay, so now the entire world facing the water crisis, The India is one of the country which is facing an enormous water crisis. With this situation, how can we produce the agricultural crop or aquaculture crop? Okay. See, in this slide, this is a serious alarming. This is a serious alarming. There are millions of farmers, they are committing suicide due to failure in their agriculture or aquaculture crops. So, according to National Crime Records Bureau of India, they reported that 13,754 farmers committed suicides in 2012. 12,360 farmers committed suicide in 2014. And likewise, every year, thousands of farmers, they are committing suicide because of their failure in their agriculture. Because their main livelihood is agriculture. If agriculture failures, how they can survive? So with the data for 2016, totally 3,33,407 farmers in India, they died by suicide in 1995. This is the data, statistical data given by government. But maybe the real data maybe is different. Okay. So at this situation, at this critical condition, as you know well, India is a country, which is the agriculture is a major employment or livelihood. So agriculture is our backbone of our country, our nation. In particular and also in the global scenario is also agriculture is the backbone of any country. But now the agriculture and aquaculture is facing failure because of lack of water. So in this juncture, in this situation, circumstance, is there any solution to produce the agriculture crop or aquaculture crop using minimum water, using minimum land, or without water or without soil? So the solution is, I am here proposing, the solution is nothing but aquaponics. Aquaponics. Okay, so some of you may be aware about the aquaponics. So let me explain about aquaponics. What is aquaponics? So aquaponics is nothing but the method of growing crabs, that is agricultural crabs, and fish, aquaculture crabs, together in a recirculating system. This is the definition for aquaponics. A method of growing agricultural crabs and aquaculture crabs, that is nothing but fish, together in a recirculating system. So this is called aquaponics. This is nothing but the old name is organic farming or traditional farming. Now it came with a fancy name called aquaponics. Okay, so with the minimum space, with the minimum water, we are growing the agriculture plant and aquaculture plant in a controlled condition. That is called aquaponics. This is the simple principle or concept behind the aquaponics. You can see here, this is the step one, where the fish is cultivated. The fish will be cultivated in a tank or pond. The water, the wastewater, which contains unutilized feed and fecal matter or excreta of fish, that water is a good source of nutrients. That water will be pumped from fish tank, it will be supplied or pumped to the plants. The plants where the plant is growing on the media bed. So as you know well, the plant, they need nutrients for their photosynthesis. So the fish wastewater is a good source of nutrients. We are just supplying wastewater, fish culture wastewater to the agriculture plant, which can absorb all available nutrients, excessive nutrients in the wastewater. And again, that waste, the water, treated water will be, once it completed, they can again come into the fish tank. So this is the simple logic, the simple concept behind the aquaponics. Okay. I would like to show this small video clipping, animation video clippings about the aquaponics, so that you can be able to understand what is aquaponics.
My name is Otto, and today I'm introducing you to aquaponics. Aquaponics is a system in which waste produced by fish supplies.
This is a simple principle behind the aquaponics. You can see here there are two beds. The one component is for to grow the fish,
Grow media stones,
and this is grow media where the plant is growing.
Siphon to regulate outflow of water. This is called an ebb and flow system. water is pumped into the grow bed, water level rises until the siphon automatically brings out all the water and the process resumes.
So you can see here the video.
And produce ammonia-rich waste. Bacteria cultured in the grow bed breaks down this ammonia into nitrites and then nitrates. Plants taking the converted nitrates as nutrients. The filtered water provides nutrients for the fish. Watch out for the second part of
So I hope you understood the principle behind the aquaponics. It's nothing but fish will be cultured in the tank. From the fish tank, that wastewater, which contains unutilized feed and fecal matter, excreta of the fish, that wastewater will be supplied directly to the crab, agriculture crab plant, where agriculture crabs will be cultivated. This is actually the symbiotic aquaponic cycle. So from the fish tank, fish culture tank, the wastewater will be supplied to the agriculture crab plant. Before that, no, there are microbes, bacteria. There are some bacteria which can convert the ammonia into nitrite and nitrite into nitrate. For example, you can see Nitrosomonas bacteria is a probiotic bacteria which can convert the toxic ammonia into nitrate. Then nitrate will be converted into nitrate by nitrobacter bacteria. The nitrate will be a good nutrient source to plants to grow. Okay, by using this nitrate, the plant will grow, and after the plant is absorbing all nutrients available in the water, that same water will be supplied again to the fish culture tank. So this is the recirculation system. So applying this system, we can use minimum water. The same water will be again and again we can use to grow the fish and to grow the agriculture crabs. So we are getting both. This is the simple components. The aquaponics, if you wish to develop, or if you wish to establish the aquaponics system, the aquaponics is not high-fi technique. It's a very low-cost technique using minimal components. For example, we need fish seed, we need a fish tank or pond to grow the fish, and there are some mechanical filter, biofilter where the microbes will be cultivated. So these microbes from the fish culture tank, the wastewater will be passed through the mechanical filter. From the mechanical filter, it will send to the biofilter where the microbes will be cultivated, where the microbes convert the toxic ammonia and nitrite into nitrate. That good water, the nitrate-rich water will be supplied to the vegetable plant or crab plant. Again, once from the agriculture, crab plant section or component, the water will be again bring into the sump where the water will be stored. From the sump or from storage tank, the water again will be supplied to the fish culture tank. So this is a simple principle or simple concept behind the aquaponics. Okay. And why aquaponics is important nowadays? What is the need of aquaponics? Okay. So as we discussed in previous slides, the aquaponics, it requires less than 2% of the water than that used in traditional farming. It's a less water usage, minimal environmental impact. That is very important. The water is recirculated in the system and no nutrient-rich wastewater is discharged into the environment. So it's an environment-friendly technique and less water usage technique, and energy efficiency is very high. For example, you can see in the aquaponics, we can able to harvest 2 crabs. 2 crabs means one is vegetable, another one is fish. So at a time, at a single operation, at a single crab, we can able to have harvest or get 2 crabs. One is vegetable crabs, another one is aquaculture crabs, fish. And another important advantage behind the aquaponics is high quality product, because here we are not using any fertilizer, chemical fertilizer or pesticide or whatever may be. We are just growing the fish and agriculture crop using organic waste. So that there won't be any residuals, there won't be any chemical or fertilizer or pesticide residual if you grow your agriculture crop using aquaponics system. It is very high quality product. And another advantage behind the aquaponics, it is locally produced. So you can it reduces the cost of transportation. So for example, in area like Coimbatore, what for what crabs the demand is there. What crab the demand is there. You can select such crab to grow in your aquaponic system. Similarly, for fish, the people for what species the demand is there, consumer demand is there. You can select such a species and you can such a agriculture plant. You can grow in the system called aquaponic, so that market demand is there. There will be any international market crisis. Okay, so it is locally produced. It is produced high quality product. It is energy efficient and it has minimal environmental impact and it uses only less water, which is need of the hour. Because as we are already facing water crisis, since the aquaponics is a system which uses minimum water for the production of fish and agriculture crab, this is the need of the hour. So if you apply aquaponics in your system, we can be able to grow 2 crabs at a time. Okay, then let us see the types of aquaponics. What are the types of aquaponics are available? Okay, there are 3 types of aquaponics system. One is nutrient film technique. Nutrient film technique, NFT. The NFT is nothing but it is very popular and versatile hydroponic system. It is a simple concept behind this NFT is ebb and flow hydroponic system. It uses a pump to deliver the fertilized water from the fish tank. That water will be supplied to the agricultural crab in a hydroponic technique. Okay, it will be cultivated in vertically as well as horizontally. Okay, so the nutrients rich wastewater from fish culture tank will be supplied, pumped using the pump to a tube, a small tubular like structure. Okay, so there you can see in this picture there is the plant is growing in the nutrient film technique. The wastewater will be supplied from the fish tank. The same water after absorbing the nutrients by plant, the same water once again it will supply to the fish tank. So the fish tank, there is a hygienic water quality condition will be maintained, and moreover the plant will also use the nutrients. This is called nutrient film technique. And another type of aquaponic is floating wrap or deep water culture. Okay, so in this system, plants are grown in sheets of styrofoam. The styrofoam sheet or even thermocol sheet you can use. Okay, you can see here in the tank, there is a water and the fish is growing inside the water. And just above the water surface, there's a thermocol or styrofoam sheet in which a small holes will be made. The small holes will be made and plant will be germinated in a small, the wrap system using a small bowl-like apparatus. Okay, so here the beneficial bacteria live in the wrap tank or throughout the system. The extra volume of water in the wrap tank provides a buffer for the fish and reducing the stress and potential water quality problems. So this is a very simple technique. You can see in this picture, by using the styrofoam boxes or thermocol, just you can make a hole, a recommended level of hole. I will tell after the suitable size of the hole in this system. And this will be grown. You can see in the root system, the root, half of the root will be in the atmosphere and half of the root will be in the water column. This is called floating wrap or deep water culture. And the last but not least, this media-filled grow beds. Media-filled grow beds is nothing but in this system, the media-based system, plants are growing in inert planting media such as gravel, rock wool, sand, or clay pellets. So using gravel or sand, okay, and the clay pellets, you can just germinate the plants on the growing bed, media bed. And here you can see in the fish will be cultivated in this tank. From this, the water will be supplied to this growing bed tank. Through growing bed, the all available nutrients will be utilized by the plant, and the treated water once again it will reach to the supply to the fish culture tank. So using gravel, rock wool, sand or clay pellets, you can just make a grow bed medium on the any container. In any container, you can use unutilized the waste material also to prepare the grow media. Okay, this is called grow media filled or grow beds culture. So with this advantage, actually throughout the world now there is a great demand for aquaponics, and the aquaponics in recent past they became very popular among the agriculturists and aquaculturists. So because of several advantages behind this aquaponics, we in our lab, we started working on developing a laboratory scale aquaponics to culture the agricultural crabs and aquaculture crabs. So now, year after, this is our own findings, our own experiment. Some of our students, they are working on this aspect. Okay. For example, we have established, see here you can see, this is the system what we have developed in our laboratory. See, this is the NFT nutrient film technique, and this is the deep water culture or floating raft system. So this laboratory scale or small scale aquaponics we have developed in our laboratory. By using this aquaponics models, aquaponics system, we cultivated the fish, shrimp, ornamental fish, edible fish, and also some agricultural crabs. So I will explain. Yeah. Actually, we took the green gram, Vigna radiata, to evaluate whether the aquaponics really is helping for agriculture production. Okay, so that we have selected the green gram, Vigna radiata, as a crab plant to grow in the aquaponics system. Okay, then here you can see this is a famous ornamental fish that Moli, Phocelia spinos. So actually, one of my project student, Mr. Stephen, he did his experiment. All this experiment he only did under the co-leadership of Dr. Nandini Devi, one of my postdoctoral scientists. Okay, so we took this fish, Moli, Phocelia spinos, is ornamental fish, and we took the agriculture crop, Vigna radiata, green gram, to grow in the aquaponic system. See, this is the experimental setup where the NFT, nutrient film technique, will be used, and simultaneously we used the deep water system or floating raft system. Okay, I'll show you the result. This is the result. So actually we made a control system. The control system is nothing but the fish culture without the agricultural crab. Okay, only fish will be cultivated. And whereas in aquaponics system, the fish and combinedly with agriculture crop will be cultivated. That is called aquaponics. So we made two methods, NFT and deep water culture. So this is the result I would like to explain. See, the fish length, the fish growth in terms of length and weight. The fish length, especially the Moli, length was 5.9 centimeter in NFT aquaponics, whereas in DWC, deep water culture aquaponics, it was 5.7 centimeter. Whereas in control system, the animal will reach only 5 centimeter length. Likewise, the weight in aquaponic system, in NFT, the weight of the animal was 3.77 gram, and at DWC aquaponics, the weight of the animal fish was 3.9 gram. Whereas at control system, the animal can able to grow only up to 3.14 gram. Okay. Similarly, we have also evaluated the survival of the fish. The survival is comparatively higher in aquaponic system, where the 95% of the survival in fish was recorded, whereas in control system, only 90% survival was obtained. And we have also evaluated the biochemical profile of the fish, which grow under the aquaponic system and control system. So the fish which grow under the aquaponic system resulting more protein, carbohydrate, lipid than the control system. You can see this is very important table. You can see here, as we are discussing about the water conservation, okay, say food production. This is a very interesting result we have got during the end of the experiment, very particularly ammonia, nitrate and phosphate. In control system on 15th day, you can see here the ammonia level was 0.09 milligram per liter. Okay. Whereas in aquaponic system, the ammonia level was 0.03 at NFT, and whereas at DWC 0.01. Similarly, nitrite concentration in control system 0.348, whereas in aquaponic it was 0.28 and 0.13 respectively. Similarly, nitrate and phosphate, you can see in nitrate 1.32 at control system, whereas at NFT 1.02 and DWC it was 0.06. Such the nutrients level is declined drastically in aquaponics system, so that it clearly indicates that if you use the aquaponics to grow your fish and agricultural plant, there won't be any excessive nutrients in your system, so that it won't affect the environment, land or water body. Okay, this is a very important findings we have got out of this experiment. Okay, so this is the photographic evidence. You can see this is the control system. This is a fish which grown under the control aquaculture system, and this is the fish which was grown under the aquaponics system. This is NFT aquaponics. This is DWC aquaponics. So it clearly shows that the aquaponics system enhancing the growth, survival, and water quality of the culture system when compared to control system. Okay, and this is the plant parameter. You can see the plant length and plant weight. So in control and NFT, you can see here the root length 3.8 centimeter in control, whereas in aquaponics it was 5.7 and 5.9. Similarly, the leaf length and stem length, okay, whole plant length, you can see here the plant length in control system, it was 13.4 cm, whereas in aquaponics, it was 16.9 centimeter and 17.9 centimeter respectively. So it clearly indicates that both fish and plant are growing well with high survival with good water quality condition. So aquaponics is maintaining good water quality. And another, one of my student, he did his MPhil in, this is a master degree. He did his experiment on aquaponics. Using aquaponics, NFT and DWC, he cultured the tilapia, fish, tilapia. Okay, Oreochromis niloticus, he cultured, and the plant what he used for growing in this aquaponics system is a millet. There is millet, pearl millet, Pennisetum glaucum, and even the mustard, the black mustard. So he has taken black mustard and pearl millet, and the fish is concerned tilapia. So our results very clear that you can see here the length of tilapia fish in control system, it was 4.38 centimeter, whereas the weight it was 1.34 gram. Whereas in aquaponic system, the length of the fish was 4.74 and weight was 2.17 at the end of the experiment. Whereas at DWC, the growth was 4.56 centimeter, weight was 1.62 centimeter. So here you can see the photographic evidence of the fish which grows under control system and aquaponic system. And we also evaluated the plant growth parameter. You can see here the total length of plant, especially the millet, the pearl millet, which grows under the control, it was 10.32 centimeter, whereas at the NFT it was 11.32. So similarly in DWC, deep water culture aquaponic system, the total length at control is 8.68, whereas at DWC it was 10.32. So it is clearly shows that both fish and agricultural crab plant, they are showing excellent growth in terms of length, weight, shoot length, root length, and leaf length, and the animal is showing very good survival with good nutritional profile. Okay, and this is actually the experimental data on black mustard. You can see here the total length of mustard plant. It was 8.74 at control system, whereas at aquaponic 8.96. Okay, similarly in DWC also. So comparatively, the plant length, weight, and leaf length is higher always in aquaponic system when compared to control system. This is, you can see here, the water quality conditions maintained in control system and aquaponic system. You can see here the dissolved oxygen. The dissolved oxygen level will be highly increased in aquaponic system. You can see here in control system it was 5.89 milligram per liter, whereas at aquaponic 6.35, 6.54. This increased dissolved oxygen level is due to the plant photosynthesis activity in the aquaponic system, which will support the animal, fish for their respiration, which will improve the growth of the animal. And likewise, you can see here the nutrient, ammonia level, it was 1.012 milligram per liter at control system, whereas at NFT 0.135, 0.128. All nutrients, starting from ammonia, nitrate to phosphate, all nutrients level is comparatively lesser in aquaponic system, whereas in control system it is very high. So it's clearly indicate that if you apply the aquaponics to grow your fish and agriculture crops, there won't be any environmental pollution, excessive nutrient pollution in the system. So this is some photographic evidence of the experimental setup. See, this is actually DWC experiment, deep water culture, aquaponics, where the mustard and millet was grown, and here the fish, the tilapia fish was cultivated. So we have got very good result with the tilapia and other agricultural crops. And another my student, my MP student, he worked on growing the shrimp, Pineus venami in aquaponic system. So here you can see the Pineus venami growth at aquaponic system. It was 11.0 centimeter in terms of length, whereas at control system it reached only 10.1 centimeter. So similarly, the weight of the animal shrimp which grown in aquaponic system, it shows 6.27 gram, whereas control animal which reach only 4.61 gram. Here you can see the photographic evidence. This is the shrimp venami which grown under the aquaponic system. This is control system. Okay, and this is he took Amaranthus viridis. It's a green. In Tamil it is called Kuppakirai. Okay, it is Amaranthus viridis greens. So greens and the shrimp. So shrimp and the greens was cultivated in the aquaponic system with reference to control system. So the plant, the greens growth also excellently obtained when compared to control. You can see here the greens length 12 centimeter, whereas at control system it was only 10.7 centimeter. Similarly, the weight of the plant weight in at aquaponic system it was 0.05 gram, whereas at control system it was 0.045 gram. So this is again the water quality parameters maintained at aquaponic system and control system. You can see here dissolved oxygen is always higher in aquaponic system when compared to control system, whereas the nutrients ammonia, nitrate, phosphate, all nutrients was comparatively lesser in aquaponic system. Here you can see, whereas in control system means there is a normal system. There is no aquaponic method is followed. So here all nutrients are always higher when compared to aquaponic system. We have also analyzed the biochemical profile of the animal, the shrimp. So the protein, lipid, carbohydrate, all nutritional parameters comparatively higher in the animal which grows under the aquaponic system than the control system. Okay, so actually so far we have discussed about the laboratory scale aquaponic system. Okay, laboratory scale aquaponic system. So now let us see a few things about the commercial aquaponics. So actually we have come out with good result in laboratory scale. Okay, but with the laboratory scale result we could not able to take to the field. We could not able to suggest or recommend for the commercial application, unless otherwise we can prove in the commercial system. So this is one of the commercial aquaponics. This is not our own result. This I have downloaded from the net. Okay, this is a typical example for commercial aquaponics. Okay, yeah, for a commercial aquaponics we need greenhouse. So we have to construct the greenhouse. Inside the greenhouse we have to develop or establish the fish culture tank, and there are different components. Okay, and this is actually a typical example for a commercial aquaponics which was built inside the greenhouse, and there are different types of aquaponic system is there. I will explain here. Yeah, this is the typical integrated recirculating aquaponic system. Okay, so this is this blue color. This all FRT means fish rearing tank. From the fish rearing tank, the wastewater will be sent to the mechanical filter. From the mechanical filter, it will pass on through the biofilter where the microbes, okay, the bacteria which will convert the organic waste, ammonia, nitrate into nitrate. That nitrate-rich water will be supplied to the hydroponics. Hydroponics means where the plant is growing. So there are different techniques, NFT, FRS and MPS. FRS means floating raft system, NFT nutrient film technique, and MPS media bed system. Okay, so all the water from the fish tank will be after filtering through the mechanical filter and biofilter, it will supply to the hydroponics. Once it hydroponics, all nutrients will be absorbed by the plant to grow their for the photosynthesis. From the hydroponics, the water will be collected in the sump. Okay, from the sump, it will again supply to the all fish culture tank. So this is a typical integrated recirculating aquaponics. Actually, this I have submitted this proposal to DST for funding to establish the commercial aquaponics system in our university campus. Hopefully, I will get it, but I am waiting for the result. Okay, so yeah, this is the typical NFT. NFT is nothing but a thin layer of nutrient-rich water flows along a tube or closed gutter into which holes are cut and the plants are placed. This is, you can see here, these holes are cut and the plants will be germinated in a small gravel, okay, using this mesh pot. This is called mesh pot. So this mesh pot along with the plant will be placed in this hole, okay, in this hole. So while supplying the new water, wastewater from fish tank, the wastewater will be slowly going. You can see here. So 10 to 15 centimeter wide. This is the tube, the nutrient tube size, and 20 to 30 meter long. Okay, such a way this NFT was developed, established. Through this, the plant will be cultured. This is commercial way of culturing plant and fish. See, this is floating wrap system. You can see here is a styrofoam box or a thermocol. So here 20 to 30 centimeter depth will be, the holes will be made. In that hole, the plant will be germinated in a plant pot. Okay, so the part of the root, the plant root, part of the plant root will be exposed to the atmosphere, and part of the plant root will be exposed to the water. So the plant can grow well. This is the what is called a floating wrap system or deep water culture system. This is media-based system. In media-based system, the plants will be growing in a medium such as gravel, clay balls, vermiculite, cinders, etc. So using the gravel instead of soil, we can use the gravel. Okay, where the plants will be germinated and the wastewater from the fish tank will be supplied to the grow bag, where the plant will utilize the nutrients. The same water will be in the form of ebb and flow. The form of ebb and flow concept, the water again will be drained to the fish tank where the fish is growing. Okay. Yeah, the plants suitable in aquaponics system. What are the plants? Not all the plants are suitable to grow in aquaponics system. Similarly, not all the fish suitable to grow in aquaponics system. There are few plants being identified to is suitable to grow in aquaponics system. So, for example, tomato, pepper, cucumbers, beans, peas, squash, broccoli, cauliflower. These are the plant identified which is suitable to grow in aquaponics system. Similarly, the fish which is suitable for aquaponics system, tilapia is a best fish to grow in aquaponics system. Tilapia is a best fish. Okay, followed by trout. This is tilapia, trout, carp, and koi carp. This is koi carp. It's an ornamental fish. And perch, bass, sea bass, perch, and crustaceans like shrimp, Penaeus, Monodon, Penaeus venami. These are the suitable fish species to grow in aquaponics system. Okay. So this is actually a small video clipping, but due to time constraint, I just simply skip this video because already now it's 3:30. Okay. The next person is online for next talk. So in general, what I would like to summarize here, based on our experiment, using the aquaponics system to grow the fish, tilapia, and ornamental fish, and edible shrimp, Penaeus venami, in combined with agricultural crabs such as the Vigna radiata and millet, mustard, okay, and greens. It is very, we have got a very good result. The aquaponics is improving the growth and the yield in both fish and agriculture crab without affecting water quality. That is very important. So in this context, the aquaponics is a good alternative. Just I would like to conclude, the aquaponics is a good alternative because it is less water consuming method of aquaculture and agriculture, and they need minimum space for aquaponics, and moreover we are able to get good nutrient rich product, high quality product. So with less volume of water, with small space, less space, we can be able to have more profit in terms of yield, crop yield, which is having very good nutrition. So there is no doubt that aquaponics is a promising alternative method for the culture of fish and agriculture crab, which can be able to supply the nutritions or cheapest protein source available on land to the human being. So that by developing aquaponics, we can be able to increase our agriculture production and aquaculture production, so that we can come out with good nutritional improvement. We can be able to supply the poor peoples, especially the peoples, those who are lacking food, nutritional food, and the water crisis. So especially with this water crisis, we are facing water crisis. So for this situation, the aquaponics is one of the promising aquaculture method. So that I can recommend that aquaponics for establishing a large scale or commercial scale aquaponics wherever possible. So I would like to give a suggestion to the policy makers. Okay, actually in India we have more than 9,000 square kilometer coastline. Okay, however the area under utilization for farming is less than 1%. Similarly, an estimated area of brackish water available for aquaculture in India is about 1.4 million hectares. Of this, about 14% has been brought under farming. We have an enormous area, coastal area. We have an enormous volume of water, seawater or salt water, which is not suitable for any purpose. Any purpose means for drinking or irrigation. So why don't we establish the aquaponics, especially the saltwater aquaponics, the coastal aquaponics along the coastal area wherever possible, so that we can grow the fish along with salt-tolerant agricultural crabs. Salt-tolerant agricultural crabs. There are some crabs have been identified. Even it can grow in saltwater aquaponics. For example, beetroot, tomato, and some agricultural crabs can able to grow in saltwater also. So that even we have seaweeds. So my recommendation or suggestions to the policy makers, okay, we can establish under the financial support or banking support. If we establish the aquaponics along the coastal area, so that we can use the arid land, the waste land for the fish production or agriculture production by adopting the aquaponics system to grow the fish and to grow the agriculture crop, which can help to eliminate the hunger, malnutrition, and the poverty of our world in general and our country in particular. So moreover, the aquaponics system is helping for water conservation, because it treating the water so that it's minimizing the pollutant, and they say since we are adopting the recirculating system, we are using minimal amount of water through the recirculation method. So my suggestion to the policy makers, the aquaponics system should be encouraged, because in India not much aquaponics, commercial aquaponics are established. To my knowledge, only two or three aquaponics, commercial aquaponics has been established so far in India. Okay, so my appeal to policy makers or government, you can support the farmers, both agriculture farmers, aquaculture farmers, to develop the aquaponics along the coastal area. That is sustainable aquaponics. I can tell you, you please note down, sustainable way of aquaponics system, which will help to fight against the hunger, malnutrition, and the poverty of our country. So I take this opportunity to thank the authorities of Nirmala College for Women, Coimbatore, for inviting me, for giving me an opportunity to deliver this talk. So the authorities who include President, Secretary, Principal, the Vice Principal, Dean Academic, okay, and the head of the Department of Zoology, and last but not least, I would like to thank the organizing secretary of this webinar, Dr. B. Dhanlakshmi Madam, for giving me an opportunity to share my experience and view with your people, with you peoples. So last but not least, I would like to thank my university authority for granting permission to deliver this talk. So with this, I will conclude my lecture. If any queries, any clarification, any doubt, I am very happy to answer to your queries. Thank you very much. Thank you all once again for time listening. Okay, now you are most welcome to raise your clarification or queries.
Thank you so much, sir, for your informative talk. Participants, if there is any queries, you can unmute yourself and ask to sir directly. Participants, you are requested to unmute yourself and ask sir directly.
Sir, very good presentation, sir. I have one question. Sir, basically in aquaponic system, the salinity is a very big problem. Like I am having some biofloc tank with me. How I can use this biofloc tank water in this aquaponic, because we are using right now as a heterotrophic bacteria for ammonia conversion, and there is some filtration type of a tank is required for that aquaponic system, or we can use directly that water to the plants and in the base.
No, sir. Before sending the wastewater to the plant, we can actually we are using the biofilter where the microbes will be cultivated, which will convert the organic waste. I mean, the toxic ammonia and nitrate into nitrate. So nitrobacter, bacteria we are recommending. So definitely the bacteria will be used to treat the wastewater before sending to the plant component.
But the wastewater in the biofloc tank is already, I think that heterotrophic bacteria take place and NPK value is high in that water.
That is the case. I no need for further biofiltration. You can directly use the further.
But the salinity is come here because salinity work, no? Because
actually you are asking about the salt water hopefully.
Yeah. Because 2 ppt salinity is there, 2 mg per liter.
Yeah. That is that actually I read an article. There are some few plants, for example, some tomato, beetroot, and some plants or agriculture, vegetable crops are able to survive in 2 ppt salinity. And I am here suggesting you, if you use the salt water for in biofloc, salt water, whether you go with seaweed cultivation, seaweeds and salt marsh plant. So nowadays there is a great demand for salt marsh, especially the California.
Seaweed required more than 14 ppt, 14 mg per liter salinity, because I am thinking for that. Now I am thinking for other things also like spirulina and other things, but this is also there is a big problem of salinity there.
Okay, and that is the case, sir. The salt tolerant rice variety is there. I hope you might be heard about the salt tolerant rice is developed by MS Swaminathan Research Foundation, MSSR.
Sir, if possible, please share that variety and from where we get that seed.
At present, I am not able to tell, but let me discuss with the scientists from MSSR, because they have developed the salt tolerant rice by transferring gene from mangrove, Abyssinia. The mangrove, just they are isolating gene, and they can transfer to the rice variety which is growing in saline soil and salt water.
Okay, sir. If I send the mail to you, you can reply for that, no, sir?
Yes,
mail ID, because in first page you have that mail ID, no, sir? In first page of the PPT.
Yes.
Yeah, okay, thank you.
Thank you, sir.
So there is a question from Mr. Priya.
Yes, madam.
Sir, there is any limitation like temperature, pressure in aquaponic system?
Temperature, pressure.
Yes, sir. There is any limitation like temperature, pressure in aquaponic system?
Actually, for everything there is a limitation. Okay, that we have to optimize. We have to optimize with reference to species which we are growing in aquaponic system. Okay, moreover, this aquaponics will be done in controlled condition. Controlled condition means greenhouses. Okay, to maintain the temperature, we are using aeration. We are giving aeration in fish culture tank, not fish water, culture tank water, so that there won't be any temperature issues. There won't be any issue, temperature issue. Even for pressure, so whatever the suitable conditions to be followed, that should be studied, optimized previously before you implement the aquaponic system. I think, to my knowledge, there won't be any issue related to temperature and aquaponics.
So another question, what type of bacteria can be used in aquaponics so that no ammonia can form?
Actually, bacillus species they are using in aquaponics as like in aquaculture pond system. So bacillus is generally a good bacteria to be maintained in the aquaponics system to convert the waste.
Okay, sir. Another question from Praveen Prasad, sir. RAS is very costly as compared to bioflux system. Can we use bioflux system water in aquaponic system? If yes, then what is the process of using this?
Yeah, there is no doubt at all. As I agreed with the participants. Definitely the RAS actually somewhat high cost technique. Instead of that, we can go for a bioflux system where the natural organisms will be maintained. For example, even now the bioflux, co-flux is coming into the system. Bioflux, co-flux, which is a novel method to grow the stream. So definitely the bioflux system is a comparatively less cost system than the RAS.
Okay, sir. Thank you so much, sir.
Thank you, madam. Is there any more question from participant side?
Any queries, participants? Sir, can we wind up, sir?
Yeah, madam. Thank you.
Okay.
Once again, I thank all the organizing team and authorities of this college for giving me an opportunity. Now I, on behalf of our Bharatdasan University and my own behalf, I wholeheartedly welcome Professor Dimitri Lajo sir.
Sir, 1 minute, sir. We'll have a vote of thanks for you, sir. Then we'll sign it, sir.
Thank you.
2 minutes, sir. Now I request Dr. Chitra to propose the vote of thanks to Dr. Sandhanam, sir. Over to Chitra.
Good afternoon to one and all. On behalf of PG and Research Department of Zoology, Nirmala College for Women, and my own behalf, extend a very hearty vote of thanks to our speaker, Dr. Sandhanam sir, Associate Professor, Department of Marine Sciences, Bharatdasan University, Trichy, for sharing your precious time with us. Sir, your presentation was very clear and informative. And in your presentation, you gave an excellent coverage to aquaponic system. I must mention our deep sense of appreciation to you, sir, for your explanation on water crisis, the type of aquaponic systems, and your laboratory aquaponics experimental setup, and about commercial aquaponics. We are very much grateful to you, sir, for sharing your research experiences and knowledge. Thank you, sir. Thank you very much.
Thank you, madam. Thank you once again for giving me an opportunity. I am very happy to meet you all. Thank you very much.
Thanks a lot, sir.
Thanks, madam.
Thank you, Dr. Chitra. Thank you so much, sir. Can I invite Dr. Mitri Lajos to join the session, the next resource person?
Yeah, hello. Do you hear me?
Yes, sir.
Do you hear me? Yeah?
Yes, sir, you're audible, sir.
Yeah. Would you like for me to start now or wait 10 minutes?
Sir, we have a welcome to you, sir.
Yeah, so you would like to start
1 minute, sir.
right now?
Yes, sir. We welcome you, then you can take up your session, sir.
Yeah.
A very good afternoon to one and all the participants. Welcome back to the second session of the one-day webinar series in Animal Sciences 2 on the topic of challenges for water resources sustenance. As Oprah Winfrey said, education is the key to unlocking the world, a passport to the freedom. Amidst of the unpleasant pandemic the world is facing, we are indeed overwhelmed by the interest and initiative taken by all the participants to imbibe valuable insights, and we hope the series will definitely give you constructive ideas in the field of research. On behalf of the PG and Research Department of Zoology, Nirmala College for Women, are profusely elated and honored to formally introduce the eminent speaker for this session, Dr. Mitra Rajus, Associate Professor, Department of Ecology and Hydrobiology, Faculty of Biology and Soil Sciences, St. Petersburg State University. First and foremost, we are extremely happy to have a dignified scientist hosting this session for us. His journey started as an undergraduate student at St. Petersburg State University, after which he has been a senior researcher at Russian Academy of Sciences, and in the year 2006 as a Fulbright scholar at the University of New Hampshire, USA. His specialization and interest in the fields of researches are population biology of marine fishes and invertebrates, population phenogenetics, stress assessment, history of fisheries, historical ecology, population dynamics, and sustainable fisheries. Sir is a peer-reviewing member of many distinguished journals, to name a few: Journal of Marine Biology, Association of United Kingdom, Environmental Toxicology and Chemistry, Journal of Biogeography, Journal of Sciences and Pollution Research, etc. Sir has guided and currently guiding undergraduates, postgraduates, and research students. He has educated many doctoral theses. Sir, as a consultant, is associated with many reputed organizations such as Marine Resource Assessment Group, MARC, American Scientific Certification System, Global Sciences, Institute of Market Ecology, Gordon and Betty Moore Foundation, Marine Stewardship Council, World Wildlife Fund, Wild Salmon Center, etc. He has published 87 research chapters, 15 read book chapters, and 4 books. In addition, with respect to various areas of specialization. Before we go over the session, we are indebted to Dr. Mitra Rajus for giving us your time to share with us your experience and research knowledge. Once again, I wish you all a convivial welcome on behalf of my department and colleagues. Thank you, sir. I will request sir to take over the session. And I wish all the participants an insightful and stimulating session. Thank you. So over to you, sir.
Thank you. Thank you very much for your introduction. And I will try to start now. Do you see my presentation?
Yes.
Yeah? Okay.
Yes, sir, we can see. Yes, sir.
It's very nice. Thank you. Thank you very much. So, the title of my presentation is The Use of Flag technique asymmetry to assess the effect of water pollution on aquatic organisms. First, I would like to make some short introduction. So, we have many problems in relation to COVID epidemic, but I think that it's a very important development is organizing webinars like that. So when we can cover several thousand kilometers just without spending any time, and you can see what's the distance we are covering right now. So I start to do with this studies on flag technique asymmetry since late 80s, and did many aspects of this technique, and have several publications on flag technique asymmetry as a method to assess water pollution. So you can see here first pages of several presentations, of several. There is some noise in
நோ மணி அவ்வளவுதான் என் காசுல 50 எவ்வளவு பெரிய காசு தரும் ஓடிப்போ.
Yeah. Okay. So you see here some first pages of my papers, and I think that in the most related to this presentation is a chapter in recently published book Fundamentals of Ecotoxicology, written by Michael Newman, and there are several research papers on this. So chemical pollution is a major challenge for the water resource sustainers, because it results in a serious direct and indirect effect of human well-being. And you can see here there are two aspects of this. So first is direct effect of impairing our health via consumption of polluted water. And the second is indirect effect, which damage ecosystem services via disruption of the structure and function. And my presentation will be devoted to the second aspect. So if to analyze historical progression of anthropogenic effect on aquatic ecosystem, we can identify several such kind of effects. First and the most important so far is fishing, then pollution, also mechanical habitat disruption, introduction of new species, and climate change. So there are 5 main ways the human impact the aquatic ecosystem, and this is this shift from the pristine ecosystem to disturbed ecosystem. And so you can see that pollution is a very important factor which affects the aquatic ecosystem. And when we there are many methods which allow to study effect of water pollution on biological system, and there are many methods. And there depend on the level of biological organization, like molecules, tissues, organs, individuals, population, communities, and ecosystem. And this aspect are different. So scientific value of information is increasing from molecules to ecosystem, because the most important thing for us is how our activities affect ecosystems, not molecules. So also cost of research is increasing when we are dealing with ecosystem. Duration of research and level of uncertainties. So it's much more difficult to study population, communities, and ecosystems than molecules and tissues. But it is much more important for us. So far, we have much more knowledge about how molecules and tissues respond to the pollution. But for us, it's much more important to know how population, communities, and ecosystem respond. So, and my presentation will be about population level. Why fluctuating asymmetry? In the 70s and up to early 90s, there were several studies which show that fluctuating asymmetry respond to environmental stress, like temperature stress, like density. So as for temperature, the asymmetry is the lowest under optimal temperature, and when it is increase or decrease above this optimal, so asymmetry is increasing. With density, asymmetry increasing when the density is growing, and the has a minimal value under the minimal density. Also, when we take other environmental stresses like pollution, in this case DDT concentration for Atheriana grunion, so we see that asymmetry is increasing, clearly increasing with increasing of concentration of pollutants. Also, asymmetry can increase with temperature, and if there's a combination of temperature and some chemical pollution, you can see a clear increase of level of asymmetry, and this is shown on lizards. And this, all this paper are from 70s, 80s, and early 90s. So flattening asymmetry increases when environmental conditions depart from optimum. The main conclusion from these studies is flattening asymmetry is a sensitive and cheap method assessing various types of stress, including pollution, in the wild populations. And if we will follow the changes of number of papers with keyword flattening asymmetry in the Web of Science database, which allow us to understand the importance of the subject, we see that in until early 90s, there are only very few papers devoted to this subject. And in late 80s and early 90s, there were several motivating studies which try to increase attention to flattening asymmetry. For instance, flattening asymmetry can be used for assessment of population state as a sensitive method of monitoring. This is Zakharov in 1989. And flattening asymmetry is a useful trait for monitoring stress in wild population. And this is citation from 1990. And during the decade of 1990s, it was a very quick growth of number of publications devoted to this subject. But later, in early 2000, there were several publications which make a skepticism in relation to flattening asymmetry as a cheap and very effective method for stress. And you see that number of publications didn't grow further, but on the other hand, it became stable. And this is actually the pattern which very typical for most of new methods. So during this 10 years, flattening asymmetry technique occupy its own and quite stable place in this area as a method for assessment of environmental stress. And now I think that it's the right time to ask what is flattening asymmetry. Flattening asymmetry is a deviation from perfect symmetry. Here you see here two shells of two layers of the shell, and from the first glance it's very similar to each other, and so they are very symmetrical. But when we take a closer view, we'll see that there are some small deviation from this perfect symmetry. You can see them quite clearly. And this is manifestation of flattening asymmetry. If to consider deviation of right minus left distribution, we see that this distribution is similar to normal. There is usually no deviation from normal distribution. And this type of asymmetry is neither genetically or is not genetically determined, and not its direction and not its level. So this is fluctuating asymmetry. There are other types of asymmetry which need to be mentioned to make it clear. So directional asymmetry. Here you see the several individuals of flatfish which have the same type of asymmetry. All they are asymmetrical and on the same side. You see that eyes on the same side of the body for the flatfish. In statistically speaking, both this R minus left minus L distribution is deviate from zero. It's not normal. It can be normal, but the mean is not zero. And both occurrence of asymmetry and its direction are genetically determined. This is directional asymmetry. Next type of asymmetry is anti-asymmetry, when the structure on one side is always larger than on another, but in different sides of different individuals. So in this case, you see several individuals of flatfish, which are all asymmetrical, so because they are flatfish, but the asymmetry is different. Some individuals have eyes on the left side, and some individuals have eyes on the right side of the body. And we have bimodal distribution of R minus left values. Now we need to, I need to show the types of symmetry, not asymmetry, but symmetry. So first is bilateral. We talked so far only about bilateral asymmetry or mirror asymmetry, when the structures are symmetrical in according to the axis. And there are several examples of that. Next type of symmetry is a translational symmetry, when the structures repeat in the space, for instance, like stones in sturgeon, or like leaves in many plants. Next type of symmetry is radial, when we have a center of symmetry in the center and several symmetrical structure around the center. You see the example. Very often in the nature we have combination of different types of symmetry in one organism. And here I presented examples of two organisms which possess each three types of symmetry, which I just mentioned. Both bilateral, translational, and radial symmetry. Nudibranch mollusks and sea lilies, so they possess also. And in the nature, most of organisms possess at least one type of symmetry, and very frequently they possess several types of symmetry. To understand more about the subject, we need to learn a bit about history and theory. Well, the first systematical study of fluctuating asymmetry were performed in 1920s by Boris Astaurov, Russian scientist, and he studied structures which develop on the base of the same genotype under the same environmental conditions, because that time it was known about genetic diversity and environmental heterogeneity as a source of phenotypic variation, and it was unknown that there are some other source of phenotypic variation. And Astaurov tried to, in his study, he tried to find such examples when he can remove both sides, both known types of phenotypic variation. He studied fluctuating asymmetry of fruit fly on the second rudiment part of the wings. He also studied segments, variation of segments of polychaetes and myriapods, because segments, they are developed, different segments of these animals, they are developed based on the same genotype and under the same environmental condition. And also he studied identical twins, so they are genetically the same, but they can, of course, develop under different conditions. But if to study some particular traits like dermatoglyphic traits on the fingers, so we can neglect this environmental difference, because this type of characters of traits, they are developed very early in ontogenesis. And the Astaurov conclusion was process of formation in an organism has some independent variation which cannot be reduced neither to genetic differences nor to direct effect of the environment. This random variation is inherited in intrinsic condition of the character formation, and then small spontaneous alteration coming from both outside, from fluctuation of the environment, and from inside, from organic structure itself. This is very important conclusion, and I try to present it in more graphical format to explain it better. So what is Astaurov concept of the random phenotypic variation? So common views in that time. So you see that phenotype of natural population, wild population. So it's caused by genotype and environment. So phenotypic variation is caused by two types of variation, genotype and environment. And in experiment, it must be if we are able to reduce genotype and environment to 0, so it will be no phenotype at all. But if we have still phenotypic variation, it is caused by residual variation because we are not able to completely reduce genotypic or environmental heterogeneity. But in fact, the experiment shows that there is some phenotypic variation, even if we take all the attempts to reduce genotypic and environmental variation. And Astaurov's idea was that it's not because we are not able to reduce this type of the source of variation, but because of presence of some special types of variation, random phenotypic variation occurring during development. So during development, we have some variation which is caused variation of phenotype, even if there is no variation in genotype and environment. And in the natural population, we have combination of all three types of source of variation, which cause the phenotypic variation of the natural population. And in more simple way, so total phenotypic variation is caused by genotypic variation, phenotypic plasticity, which is related to heterogeneity of environment, and the genotypic is caused by parents variation, and also random variation of lactic asymmetry, which is caused by developmental instability, which I present you a bit later. What is contribution of lactic asymmetry of random variation in total variance? It is sometimes it's surprisingly high. For instance, in this case, in some characters on small copypod acanthocyclops signifer from Baikal Lake, so we see that random variation, which is caused by lactic asymmetry, actually. So it can you see that it can be can occupy up to half or a bit less than half of the total variation. And there's other example on toadfish, on bone structures of toadfish, and you can see that it's also quite high. If to summarize all the literature data on contribution of lactic asymmetry to total variation, we can get quite high figures. For meristic traits, it's about half of variation, and for morphometric traits is a bit less, but still quite significant. What are the causes of fluctuating asymmetry? Inability, the general cause is inability of organism to produce precisely the same phenotype under the same environmental conditions. This is developmental instability. And have a look on the visual model of developmental instability. So, if we imagine such a situation, which is actually unrealistical when the developmental instability is absent, so we can see that from the route from genotype to phenotype, we have and with development trajectory for left and right structures, they are absolutely the same, and left and right structures, they are there's no difference between them. So and there is no fluctuating asymmetry; it is zero. But in realistical situation, when developmental instability is present, the developmental trajectories for right and for left structures are different, and because of that, we have a difference between left and right structures, and we have a fluctuating asymmetry more than zero. Why fluctuating asymmetry is a measure of environmental stress? So, with increase of level of stress, resources for homeostasis, except stress response, they are decreasing because organism, they have more or less constant amount of resources which are sharing between resources available for homeostasis and resources available for stress response. And with increasing level of stress, we have organism need to allocate more energy, more resources for homeostasis, for response to stress. So with increasing stress, more and more resources, more and more percentage of resources goes to the response to stress. And because of that, resources available for morphogenesis, they are increasing, and entropy of the morphogenesis is increasing, and it increases also random variation and the fluctuating asymmetry of morphogenesis. So under stress condition, there is an alteration of morphogenesis and the increase of developmental instability. It is because fluctuating asymmetry can be used as an environmental stress measure. Now about practical aspect of use of fluctuating asymmetry for stress assessment. So fluctuating asymmetry has some advantages and some disadvantages in comparison with other methods of stress assessment. First are advantages, so applicability to the wide range of organisms, as almost all organisms have different types of symmetry. So there is it means that they have also different. They have also fluctuating asymmetry, deviation from this perfect symmetry. So asymmetry is relatively easy and cheap to measure, but there are some statistical issues which you need to address. There is a possibility for applying non-invasive techniques. In some types of characters, there are quite good possibilities for that. In some other, it's not very good. It's a permanence of response. So the fluctuating asymmetry is response to in any stage of ontogenesis permanently during the life history of the animal. So there is a clear definition of ideal state. It's absence of asymmetry. So asymmetry is zero. It's never happened, but we know that it's ideal stage. The main disadvantage of this technique is a non-specific to anthropogenical stress. So it is a general measure of stress, like other general indicators. So we can't say what kind of stress was applied when we get the results on asymmetry. We can only say that stress is applied, and then try to interpret to understand what kind of factors which are important. What characters can be used for fluctuating asymmetry analysis? It's a very wide range of characters. I worked quite a lot with bone structure on fish, and here you can see the white sea, the bone of the white sea herring with a fat lacunes, which are convenient characters to study. And in this picture, you can see several structures of salmon, of Atlantic salmon, different bones with asymmetry, with variation in seismosensoric pores, number of teeth, and various distances between different landmarks in bones. And here you see an example from three-spine stickleback, which we work with quite a lot right now. And you see that the characters are linear distances between landmarks, and we compare them from left and right, and in this way we can estimate fluctuating asymmetry. Sometimes from one animal we can take maybe 40, 50 characters, sometimes less. Also, fluctuating asymmetry can be used on some coloration patterns, like in Salmatrutta, brown trout, number of spots, for instance, or number of stripes and distance between stripes and different fish species, and not only fish, of course. In isopod Saduri entomel, we use number of theta on legs, and it's quite convenient and quite effective characters, I have to say. In copypods, we use different lengths of different body structures, caudal rami, and different theta, and antennal, and so on. In blue mussel, we use measurements of the internal structure of the shell, from the left and right. In other modules in COCL, we use also some measurement which describe the shape of the shell. It is also we did some studies on plants. This is seagrass Halophila ovalis, and in this case, the characters is a number of interception between different veins, lateral veins and the central veins and lateral veins. And you see that, for instance, in this particular case, in the left we have 12 interception, and on the right we have only 10 interception because of fusion of veins and fusion and separating of some veins. So, and this is characters which can be used for asymmetry as well. This is a scanned picture of the leaf. In other seagrass, Zostra capricorna and Zostra marina, we use different types of characters because they possess translational asymmetry, and characters was distance between septa. You see several septa, and so you can measure distance between them and compare with the ideal picture when there is the distance between septa are equal. And fluctuating asymmetry is a variance of this measures, and this is it's also a way to measure fluctuating asymmetry. In bladder fucus, we measure dichotomical branches from. They are not left and right, so they are just dichotomical, but they can be measured and used for fluctuating asymmetry. And also, I show you some pictures which I didn't talk with, but I think that they are very effective and very promising traits for study fluctuating asymmetry. In filamentous macroalgae, departures from translational asymmetry, and you see that you can measure distances between the septa. And in diatoms, there is quite a lot of different types of symmetry which you can use for studies of fluctuating asymmetry. And I never work with this kind of organism, but I have to say that due to short life cycle and the morphological features, diatoms, macroalgae, and other plants and animals are a very convenient model for environment monitoring using fluctuating asymmetry because they develop very quickly, and it takes only a few days to develop the whole structure, and therefore to analyze the effect of, in our case, water pollution on asymmetry. What are the main challenges of fluctuating asymmetry analysis? So first is measurement error, then association of individual asymmetries with size, departures of distribution of R minus L values from normality, departures from concordance of fluctuating asymmetry response across strides, and the separation of effect of different factors under natural condition when many factors act at the same time. And I will not talk about second, third, and fourth, but I will talk a bit about first and the fifth challenges. Measurement error, it manifests itself when different measures of the same specimen result in somewhat different results. And these are caused by instrumental errors, inconsistency of taken measurements, and it is because it's impossible to take measurements absolutely precisely. As fluctuating asymmetry is often very small, measurement error may be comparable or even higher than true fluctuating asymmetry. True means biologically, which is a biological property of organism. So normally when we measure some character, we have observed fluctuating asymmetry which includes true fluctuating asymmetry and measurement error. It is impossible to avoid measurement error, but it's possible to reduce it and accounted for by replication measurements or counts. For instance, we can it's another way to account for measurement error is not to measure samples in specimen by specimen, not sample by sample, but in the random order, mixing different specimens from different samples. And it will not reduce measurement error, but it will equalize it in different samples. And one common way to reduce measurement error is to select traits with relatively high fluctuating asymmetry. So it is relatively high means that it will be low measurement error or high fluctuating asymmetry. So the most important is to have high fluctuating asymmetry in relation to measurement error. Main difficulty of interpretation of fluctuating asymmetry data in the wild is that asymmetry is a general measure of stress, and it is affected not only by factors which we are interested in, but also by other factors which are which often not controlled by the researchers. And this is both advantage and disadvantage, as I thought. And based on fluctuating asymmetry, we can rank level of stress the population experience, but it is not easy to identify the factor which causes the stress. And I have to say that it's not only a problem or issues of fluctuating asymmetry as a method of stress assessment, but also the problem of all other general measures of stress. And here I show you some examples from my studies. Here's an example of fluctuating asymmetry study on smooth toadfish, Tetrapterus glaber, in Australia. We compare fish from clean estuary and with more polluted estuary where city of Sydney is situated, and we found that we have a higher fluctuating asymmetry as expected actually in polluted estuary. And you see here the actual results with clean and more polluted areas, so they exhibit higher fluctuating asymmetry. But it's in this particular study, I would like to pay your attention that we found response of fluctuating asymmetry only on organic pollution, but not on heavy metal pollution. So the response of asymmetry can be different for different types of pollution. The other example from a study in Hong Kong, it's on barnacles, which are littoral animals living on different part of littoral. And here we collected them from upper littoral, medium littoral, and lower littoral. And so we compared two clean sites and two polluted sites, and found that indeed the concentration of synchron and copper is different in tissues of barnacles, high in polluted sites. But when we studied fluctuating asymmetry, so we didn't find any differences between them. We found difference between upper littoral, medium littoral, and lower littoral, because it is in lower littoral conditions are more stable, and fluctuating asymmetry is always lower. But we didn't find difference between clean and polluted sites. And we try to understand this and think about that. We studied activity of proteinase in the tissues of barnacles, and found that in polluted sites, activity of proteinases is higher than in clean site, which means that they have a better feeding condition there, and which is so it's logically, because in polluted sites with more human impact, they have pollution on the one hand, and on the other hand they have also some discharge of the sewage, which is for barnacles, this is a source of food. So the conclusion for that is barnacles from upper littoral zone manifest higher asymmetry. Low increase of symmetry is associated with heavy metal pollution, and the effect can be compensated by better nutrition situation in the polluted location. Another quite similar situation was on our study on flagonic asymmetry in seagrass, Zoster capricorn and Halophila ovalis in Australia. We compared seven locations in the Lake Macquarie, and while one location was highly polluted because of smelt activity. So we found much higher concentration of heavy metals in sediments and tissues of seagrass, but again, no difference in flagonic asymmetry of seagrass in both species of seagrass. And we also measured concentration of nutrients, nitrogen and phosphorus, and found that it's much higher than in the polluted location. So we have a hypothesis that it's again absence of response to flagonic asymmetry is caused by the superimposition of different factors. And we also made the experiment when we grew Halophila ovalis, seagrass Halophila ovalis in different under different concentration of heavy metals, with plumbum and lead and copper. So, and you see that there's a clear response of the flagonic asymmetry on pollution. Under higher pollution, we have higher flagonic asymmetry as it expected, but we didn't find this in the wild, and probably because of superimposition of different factors. And the conclusions of my presentation is like that. The flagonic asymmetry is a manifestation of random phenotypic variation, which represents the third type of phenotypic variation standing on equal footing with genotypic variation and phenotypic plasticity. Random variation is caused by developmental instability, which characterizes ability of an organism to produce the same phenotype in the same environmental conditions. And under stress, shortage of resources result in alteration of morphogenesis and to increase of developmental instability. Flagonic asymmetry is a stress monitor. It is, as a stress monitor, is applicable to the wide range of organisms. It's relatively easy and cheap to measure, and can be used non-invasively, has permanent response to environmental stress and a clear definition of ideal state. At the same time, being a general measure of stress, the data on flagonic asymmetry in natural condition require additional information for correct interpretation of the acting factors. And currently, after 3 decades of intensive studies, flagonic asymmetry occupies a stable and important place among other techniques of biomonitoring of environmental stress, and in particular for study of pollution. And I would like to thank you for inviting me to this webinar, to all the organizers, and insist on Professor Santana and Professor Dhanalakshmi, and for your attention. Thank you very much.
Thank you, Professor. Thank you for your informative talk. Participants, now you can unmute yourself and ask Professor any queries if you have. Dear participants, if you have any queries, you can ask directly to Professor by unmuting yourself.
Professor?
Yeah.
Hello. Prof, I have observed ossification happening in a spinal cord of a fish. Can you hear me? That is happening with the same species repetitively. I have observed the X-rays of the concerned species in several stages. Can that be a form of asymmetry? The fish species is Drepani punctata.
Excuse me, repeat please. So it's not very good connection.
Can you hear me now?
Yeah, I hear you. Yeah. Yeah.
There is a fish species, Drepani punctata, and
Yeah.
I have taken X-ray of the fish in several stages: juvenile fish, adult, and following several other stages. And I have observed ossification in the fish bone, a swelling in the fish bone.
Yeah.
And morphologically, externally, do not happen to show any change. But ossification is
change between
can that
changes
be
between different
changes
excuse
between
me, okay, yeah.
Yes.
Changes between different ontogenetic stages, or what kind of changes?
See the skull. See, when I consider Drepani punctata and Drepani longimena, which are very close fish species, the skull shape in the fish has a drastic change. In X-ray, when I have taken X-ray of the fish, the skull shape of the fish in both the fishes of Drepani punctata and longimena have changes in the arrangement of skull bones. That may be due to the swelling of the spinal rib. Can that be counted as asymmetry? Or
you need to study it, so it's not very difficult to study because quite many studies of
okay, Professor.
Yeah, it can be due to asymmetry, but it's needed to be studied, especially. Yeah.
Yes, Prof. I can get your contact details and get back to you through mail. Thank you.
Yeah, of course, you are very welcome to contact me, and we can discuss this in more details than now. Yeah, of course.
Any more queries from the participants?
Madam, there are some questions in a chat box. There are some questions.
Yes, sir.
You can answer.
Professor, there is a question. What is the impact of global warming and climatic change on fluctuation asymmetry?
Yeah.
What?
In general, so, I hear you. So it's a question about the impact of global warming on fluctuating asymmetry.
Hello, sir. Good evening.
Yeah.
Can you hear me?
Yeah.
So what are the changes in the field, especially in asymmetric, during the eclipses in the water reaction?
Say it again, please.
So what are the reactions during eclipses in the water, sir, asymmetrical animals?
Excuse me, I don't understand you. So it's the connection is not perfect, I would say.
Yeah, Miss, I think Miss Haripriya, you please post your question in the chat box so that the organizer will raise this question. The resource person can able to answer because there is some interruption in the net connection.
Dhanalakshmi?
Yes, madam.
Please raise the question. Please raise the question from the chat box. From the chat box, there are questions. Please.
Yes, ma'am. Yes, ma'am. Professor, is there any particular type of stress that can affect both phenotypic and genotypic changes in the aquatic organism?
Excuse me, I can't understand. So it's the sound is not very clear for me. Yeah,
okay, let me tell you once again, Professor. Is there any particular type of stress can affect both phenotypic and genotypic changes in an aquatic organism?
Yeah. Repeat, please. So it's I can't.
Can you hear me, Professor?
Yeah, I can hear you, but it's the noise. The sound is not very clear.
All right, Professor. Then I'll go for the next question. If you can please tell me the answer. What is the impact of global warming and climatic change on fluctuation asymmetry?
Yeah, the impact of global warming and climatic change on asymmetry is like the impact on any general measures of stress of organisms. So for some population, which are close to the border of the distribution range. In India, for instance, when they're very close to the high temperature level in the tolerance, so the impact of global warming will be negative because it will shift them beyond the tolerance level. But for instance, in Russia, in some parts of in the White Sea where I work quite a lot, it's the many organisms live on the northern border of the distribution range, and because of that, the warming will improve the situation, the environment for their life. You understand me?
Yes, Professor. The next question is.
Yeah. Okay.
Can I move to the next question, Professor?
Yeah. I'm ready.
Different antigen and physical structure change with pollution. Did genes also change? Can you?
What structure changes pollution?
Different antigen and physical structure change with pollution in organisms, isn't it, Professor? Will genes also change? There was a question by an audience.
Yes, of course. But in this case, we compare population. Yeah, we compare population which are which are very genetically very close to each other, and in this case, we can attribute the changes in they to environmental conditions, not to genetic, of course. Yeah. So in generation, during many generations, of course, these changes can be due to genetical changes, but within one generation, so they are related to environmental change.
Professor, the next question from one of the participants: can any asymmetric fluctuation change into any adaptive character? If yes, give an example, sir.
Yes, of course. Each character has fluctuating asymmetry, but in adaptive characters, so it is more difficult to measure it because it is usually quite small, and it is very, very difficult to see biological signal. And because of that, very often for analysis of asymmetry, researchers take the not adaptive characters, which are not very important for survival of animals. And because of that, so it's easy to see the asymmetry. And usually on asymmetry on different types of characters is manifested concordantly. So if the in polluted areas, for instance, asymmetry is higher in different types of characters, in all types of characters. So, but it is more easy to study it on not adaptive characters or not very adaptive. So, for instance, wings in Drosophila or in birds, so they have very, very low asymmetry, and it's very difficult to study it. Yeah.
Thank you, Professor. Another question: can ossification cause asymmetry? Can that be a stress stimulation?
Ossification.
Yes. Can ossification cause asymmetry? Can that be a stress stimulator?
It's difficult to tell for me this, because ossification is a developmental process, and why? So I think that it's both asymmetry and ossification are caused by the same factor. I don't know which kind of factor is this. Maybe pollution, maybe some. I don't know. So some other factor which caused by asymmetry and ossification. But I don't think that ossification itself can result in increase of asymmetry.
One more question, Professor. Which type of pollutants can affect the asymmetry in an aquatic organism?
Which type of pollution?
Pollutants.
Any pollutants which cause stress for organism can affect asymmetry as well. So any pollutants, they are pollutants by definition. If they are pollutants, they affect stress, and they cause stress, and because of that they affect asymmetry.
Next question, Professor. What are the changes in the aquatic animals during the solar eclipse?
During what?
Solar eclipse.
I did not get the last word.
Solar eclipse.
Solar?
What are eclipse?
I didn't understand, sorry.
A change in the movement of the sun, Professor. If there is any change in the movement of the sun, what are the changes you can express in the aquatic animals during that solar eclipse?
Solar radiation, yeah?
Yes, Professor.
Yeah. So, you know that water is a very good protection for the solar radiation. But for instance, in one example from my study was on barnacles from the upper part of littoral. So, and it means that in the low tide they are exposed to solar, to sun quite a lot. And in this case, in our study, we found the higher symmetry of animals in the upper part of littoral. And maybe in this case, this higher symmetry is caused by solar radiation. But to say it precisely, what kind of stresses cause flactonic asymmetry, we need to do some special study, like experimental study or something like that. Yeah, if it is stress, yeah, of course, it can cause asymmetry.
Thank you, Professor. Participants, if there is anyone who can unmute yourself and ask the queries directly to Professor. Ma'am, can we wind it, ma'am? Ma'am?
Yes, Dhanalakshmi.
Can we wind it, ma'am?
Yes.
Ma'am.
Yeah.
Now I kindly request Dr. Rosalyn Mary ma'am to propose the vote of thanks to Professor Mithai Lajos.
You are precious in the eyes of the Lord. Good evening to everyone. First of all, I thank the Almighty God for the success of the webinar series in Animal Sciences 2 on the topic Challenges for Water Resources Sustenance, organized by PG and Research Department of Zoology, Nirmala College for Women, Coimbatore. I am happy to propose a vote of thanks to the eminent speaker, Dr. Dmitry Lajos, Faculty of Biology and Soil Sciences, St. Petersburg State University, Russia. In his presentation, he explained why fluctuating asymmetry with environmental factors like pollution, some of the chemical concentrations of DDT, temperature and pollution, types of asymmetry and symmetry, challenges of fluctuating asymmetry analysis, etc. Thank you very much, sir, for the enlightened talk. Thank you. My sincere thanks to Dr. Sandhanam, sir, for the informative talk. Thank you, sir. My sincere thanks to our college secretary, Reverend Sister Dr. Ruby Alangara Mary, our beloved principal, Reverend Sister Dr. Helen, for her constant support and encouragement. Thank you very much, sister. My heartfelt thanks to our vice principal and our colleague, Reverend Sister Dr. Mary Fabiola, for her prayers and wishes. Thank you, sister. I thank the Controller of Examinations, Reverend Sister Dr. Imelda Mary, for her encouragement and wishes. My sincere thanks to the Deans and IQAC Coordinator and HOD of our department, Dr. Pauline Vasanthi Joseph, for her constant support and encouragement to organize the webinar series. Thank you, Vasanthi. My sincere thanks to all the participants, staff members from various colleges and universities, research scholars and students. Without you, there is no possibility to conduct the program. Thank you all for your cooperation. My sincere thanks to all my dear colleagues for organizing this webinar. Thank you all once again.
Thank you, Roslyn ma'am. Thank you so much.
Thank you.
Thank you, ma'am.
Dhanalakshmi.
Dear, yes, ma'am.
Lakshmi, I would like to thank Dr. Dimitri Lajus for his very informative talk. And I see such a lot of work and research is going on the fluctuating asymmetry in animals. But I'm very sorry to say that I could not listen to the later half of your talk because I don't know, something has happened in this Google Meet. And I was actually very interested in the answers that you would give for the questions that were posed to you. But unfortunately, I could not listen to the answers that you have given. Perhaps I will look at the recording and see what are the answers you have given, sir. It has been very wonderful, sir, having you with us in this webinar series. And I also thank Dr. Sandhanam from Bharatidasan University, who has obliged us in times like this. And we thank you very much, sir. Dr. Sandhanam is there? No, if he's not there, I will just pass on my wishes to him. Yes, sir. Yes, sir. Thank you. Yes, sir. Yes, thank you very much for your very informative and illustrative PPT, sir. It was very wonderful. And aquaponics is something that we always like to start in our college. But I was just wondering whether it would be cost effective for us to do an aquaponics.
Definitely, it is cost effective, madam. We can establish. So if we can do jointly, we can
yes, sir. Yes, sir. That is very nice. We could collaborate with you, sir, and have one unit in the college, sir. Actually, we were planning for it, sir. And it was very kind of you, sir, to have invited your colleague and your friend, Dr. Lajus, to be here. From the questions that have been posed to both of you, I see that the participants are very much interested and they were involved in your talk. So thank you very much, sir. Thank you, Dr. Sandhanam. Thank you, Dr. Lajus, for being with us here today. Thank you so much, sir. Thank you.
Thank you.
Thank you, ma'am. Thank you. Thank you, sir. And thank you, Professor. Thank you for joining us on this day. Thank you so much, sir.
Ma'am, Dhanalakshmi, ma'am?
Yes.
I want to speak.
Yes, sir.
More webinar required regarding the aquaculture, things of new technology regarding biofloc, rash, aquaponic, and new system of bacteria, how we convert the ammonia to protein. This type of webinar, if you are conducted, that is good for all of us.
Sure, sir. We'll try to conduct it in the future.
Yes, sir. I would like to add, Mr. Praveen, sir.
Yes, sir.
Really, your suggestion is welcome. Actually, we are planning to organize the webinar on sustainable aquaculture practices, which include ROS, bioflag, co-flag, which is a novel technology, co-flag, and IMTA, raceways, cage culture. So very soon we will maybe join with Nirmala College.
Sir, I am very thankful. I am very thankful because basically I am in Delhi. Through some reference, we know that website and we know that this training is going. Because this type of things we required.
Okay. Thank you, sir. Definitely.
Personal mail. Because data is there, then you people can send that this type of webinar is coming. We have to again and again search where the webinar is going on regarding the aquaculture. This is the major problem is coming right now.
Very soon you can get another webinar.
Praveen.
Okay, sir. Okay.
Praveen, sir, coming 29, there is one more webinar conducted from our organized by our department. This is on aquaculture sustenance. You can join us in that also, sir.
Okay.
Yes, sir. We'll send you the invitation, sir. Thank you, sir. Thank you, Sandhanam, sir. And thank you, Professor, for joining us today. Dear participants, the feedback link is posted in the chat box of both YouTube link and in the Google Meet. You can fill in the feedbacks. Sandhanam, sir.
Yes, ma'am. Yes,
sir. Sir, can we